All right, so today we're taking a deep dive into something pretty fundamental.
Yeah, definitely fundamental.
It's all about how life is organized out there in the world, in all these different ecosystems. Like, have you ever thought about how how many different types of organisms share a single space?
Oh, absolutely, yeah. And not just how many types, but how many individuals of each type.
Exactly. And that's where the idea of populations comes in.
Right, a population that's basically a group of individuals from the same species,
Uh-huh.
all living together in the same area.
Exactly. And they're not just hanging out together. They're interacting, like competing for resources, maybe reproducing, all that jazz.
It's kind of like, think of a school of fish, all swimming together, or a colony of ants, all working together in that anthill.
Perfect example. And, for you IB biology students out there, this is a core concept.
Oh yeah, big time.
So in this deep dive, we're going to break down all the essentials about populations and communities, what they are, how they work, all the factors that influence them.
And how scientists actually study these things out in the real world.
Exactly. So we'll go over, the very definition of a population, because it's actually a little bit more nuanced than you might think.
Oh, for sure.
We'll then look at how scientists estimate population size. Like, imagine trying to count every single fish in a lake. That's kind of crazy, right?
Way too much time on your hands if you try that.
Haha, yeah. So we'll discuss the clever methods they use to get around that. Then we'll delve into the concept of carrying capacity, basically, the maximum number of individuals that a particular environment can support.
Right, because resources are limited, food, water, space, all that good stuff.
Exactly. We'll also explore all the different types of interactions that happen between organisms, both within the same species and between different species.
Because those interactions play a huge role in shaping communities and ecosystems.
Totally. And finally, we'll even touch on some of the statistical tools that scientists use to understand these ecological relationships.
Yeah, like the chi-squared test, which sounds intimidating, but we'll make it super clear.
So let's jump right in. First things first, defining a population. It's not just any random group of plants or animals, right?
Our sources emphasize a couple of key things here. A population is made up of individuals that belong to the same species.
Okay, so same species, that makes sense.
And they have to be living and interacting in a particular area, at a particular time.
Right, so it's specific to a place and time.
Exactly. And here's the really important part. They have to be capable of interbreeding with each other.
So they can reproduce and have offspring together.
Exactly. And generally, they're reproductively isolated from other similar groups.
Okay, so they're kind of like their own little breeding unit.
Precisely. And a great example from our sources is the emperor penguins in Antarctica.
Oh, yeah, those adorable waddling birds.
Right. They live in this really specific, harsh environment, and they primarily breed with other emperor penguins in that same area.
So they're geographically isolated, and that kind of keeps them as a distinct breeding population.
Exactly. It's this combination of sharing space, interaction, and the ability to interbreed that makes it a true population in the ecological sense.
Got it. Now, if we want to understand how a population is doing, like is it growing, declining, stable, we need to know how many individuals are in it, right?
Absolutely. Population size is a fundamental piece of the puzzle.
But it's not always easy to figure out. I mean, can you imagine trying to count every single blade of grass in a field?
Uh-huh. No way.
Mm.
Our sources actually point out several challenges when it comes to determining population size.
Like what?
Well, for many populations, the sheer number of individuals is just massive. It would be practically impossible to count them all accurately.
Okay, that makes sense. Especially if they're small and all over the place.
Right. Plus, think about organisms that live in places that are hard for us to reach, like the deep ocean or underground burrows.
Oh, yeah, those are tough spots.
And then there's the issue of mobility. Like, how do you count a flock of birds that's constantly on the move?
They'd be long gone by the time you finished counting.
Exactly. And even if you could somehow count every individual, it would take an incredible amount of time, effort, and resources. Not very practical.
Definitely not. So what do scientists do instead? They can't just give up, right?
Right. They've developed some pretty clever methods to estimate population size without having to count every single individual.
Okay, spill the beans. What are these methods?
Well, there are two main approaches we'll discuss. One is random sampling, which is particularly useful for sessile organisms, meaning organisms that don't move around much.
Like plants, right? Or barnacles attached to a rock.
Exactly. The idea is to get a representative sample of the entire area without having to survey every square inch.
Okay, so it's like taking a few snapshots and using that to estimate the bigger picture.
Precisely. And one of the most common techniques is quadrat sampling.
Quadrat sampling, that sounds kind of technical.
It's actually pretty straightforward. Researchers basically take a square frame, called the quadrat, and they randomly place it within the habitat they're studying.
Okay, so it's like dropping a frame down randomly.
Right. And then they count the number of individuals of the species they're interested in that are within that quadrat.
So they're just counting within that little square.
Yep. And they do this for a number of quadrats, all randomly placed. Then they calculate the average number of individuals per quadrat.
Okay, so they're getting an average density.
Exactly. And finally, to estimate the total population size, they multiply that average density by the total area of the habitat.
So if they have a certain number per square meter and they know the total area is, say, 100 square meters, they can multiply to get an estimate.
Exactly. Our source gives a nice example with mangrove seedlings.
Mangroves, those are the trees that grow in salty water, right?
Yep, along coastlines. So let's say researchers are studying mangrove seedlings in a particular area. They use 1 square meter quadrats, and they find an average of 12.4 seedlings per quadrat.
Okay, so 12.4 seedlings in each of those squares on average.
Right. And let's say the total area of that mangrove forest is 1 hectare, which is 10,000 square meters.
Okay, so they have the average density and the total area.
Yep. So they multiply 12.4 seedlings per square meter by 10,000 square meters, and they get an estimated total of 124,000 seedlings in that forest.
That's a lot of seedlings.
It is. And the key here is that those quadrats are randomly placed.
Right, because if they purposely put the quadrats where there were tons of seedlings, it wouldn't be a fair representation of the whole area.
Exactly. Randomness helps to reduce bias and ensure that the sample is truly representative of the entire population.
Got it. Now, what about those organisms that don't stay put? You know, the ones that are always on the move.
Ah, that's where the capture, mark, release, recapture method comes in.
That sounds a bit more adventurous.
It is. It's used for mobile populations like birds, fish, insects, things like that.
Okay, so how does it work?
So first, researchers go out and capture a sample of individuals from the population.
Okay, so they round up some of the animals.
Right. And let's say they capture M individuals in this first capture event.
For mobile.
Haha, you could think of it that way. So they capture these M individuals, and then they mark them in some way that's harmless, doesn't hurt them.
Like a little tag or a band on their leg or something like that.
Exactly. And the marking has to be something that'll last for a while, not rub off or fade away too quickly.
Right, because they need to be able to recognize the marked individuals later.
Exactly. So after they're marked, they're released back into their habitat.
Okay, back to their normal lives.
Right. And then they allow enough time for those marked individuals to mix back in with the rest of the population.
So they need some time to, spread out and mingle.
Precisely. And then they go back and capture a second sample of individuals.
Okay, round 2 of catching.
Yep. And this time, let's say they capture a total of N individuals.
N for, second capture.
Sure. So they have this second sample of N individuals, and they carefully check to see how many of them have the mark from the first capture.
So they're counting how many are recaptured.
Exactly. And let's say that number is R for recaptured.
Okay, so we have M for the first capture, N for the second capture, and R for the recaptures.
Yep. And with those 3 numbers, they can use a formula called the Lincoln Index to estimate the total population size.
Okay, Lincoln Index. Sounds fancy.
The formula is pretty simple. It's population estimate equals M times N, all divided by R.
So number from the first capture times the number from the second capture divided by the number of recaptures.
Exactly. So let's say in our example, the researchers initially caught and marked 45 turtles. That's our M. Then in the second capture, they caught 50 turtles. That's our N. And out of those 50, 15 of them had the mark. That's our R.
Okay, so if we plug those numbers into the formula, it's 45 times 50 divided by 15.
Right. And that gives us an estimated population size of 150 turtles.
Cool, so that's how they use the Lincoln Index to get an estimate.
Exactly. But there's a catch. This method only works if certain assumptions are met.
Oh, there are always assumptions in science.
Right. And if these assumptions aren't true, the population estimate might not be accurate.
Okay, so what are these critical assumptions?
Well, first, the marking shouldn't affect the survival or behavior of the animals. Like, If the mark makes them more visible to predators, that would mess things up.
Right, it needs to be like they were never marked in the first place.
Exactly. And the mark can't wear off or fall off before the second capture.
Okay, that makes sense.
Another important assumption is that the marked individuals mix randomly back into the population.
So they can't all just hang out together.
Right. They need to spread out and be evenly distributed.
Okay, so it's like shuffling a deck of cards.
Perfect analogy.
Mm-hmm.
And finally, the population needs to be closed during the study period.
Closed, what does that mean?
It means that there's no significant immigration or emigration happening.
So no new individuals coming in from other areas and no individuals leaving the area.
Right. And there shouldn't be a big difference in the number of births or deaths between the two capture events.
Okay, so basically the population needs to be relatively stable during the study.
Exactly. If those assumptions hold true, the Lincoln Index can provide a pretty good estimate of population size.
Got it. Now, we've talked about how many individuals are in a population, but what determines how big a population can actually get?
Right. That's where the idea of carrying capacity comes in.
Carrying capacity, it sounds like there's a limit to how many individuals a particular environment can support.
Exactly. Our sources define carrying capacity as the maximum population size that an environment can sustainably support.
Sustainably, meaning over a long period of time.
Right. It's not just a temporary peak. It's the level that can be maintained given the available resources.
Okay, resources, that makes sense. Like if there's only so much food, water, and space to go around, there's a limit to how many individuals can survive.
Precisely. Those are the limiting resources we're talking about. Food, water, space or territory, nesting sites, shelter, all those essential things.
So the carrying capacity is kind of like the ceiling for population growth.
Exactly. And it's not always a fixed number. It can fluctuate based on changes in the environment.
Like if there's a drought, there might be less food available, and the carrying capacity would go down.
Exactly. Or if there's an increase in predators, that could also lower the carrying capacity for the prey species.
Okay, so carrying capacity is dynamic. It can change.
Yep. And it's an important concept to understand because it helps us predict how populations might change over time.
Now, our notes also mention something called density-dependent factors. What are those?
Density-dependent factors are things that influence population size based on how crowded the population is.
So the more individuals packed into a certain area, the stronger the effect of these factors.
Exactly. And these factors often create negative feedback loops, which help to regulate population size and keep it around that carrying capacity level.
Negative feedback, meaning they sort of push back against population growth when it gets too high.
Precisely. It's like a self-regulating system.
Okay, so what are some examples of these density-dependent factors at work?
Well, competition is a big one. As population density increases, individuals have to compete more intensely for those limited resources we talked about earlier.
Right, more mouths to feed, more bodies needing space.
Exactly. So the more crowded it gets, the tougher the competition for food, mates, nesting sites, territory, all that stuff.
Makes sense. So competition ramps up as the population gets denser. What else?
Predation is another important density-dependent factor. Predators tend to focus on prey species that are more abundant.
It's like a buffet for them, right?
Exactly. If there's a lot of prey around, it's easier for predators to catch them, so they have more success and their population might increase.
But then if the predator population gets too big, they'll eat more prey, causing the prey population to decline.
Right. And that, in turn, might lead to a decrease in the predator population because there's less food for them.
Ah, so it's like a seesaw effect, with the predator and prey populations influencing each other.
Precisely. Our source mentions the classic example of the lynx and the snowshoe hare. Their populations go through these cyclical fluctuations, with the lynx population lagging behind the hare population.
So when there are lots of hares, the lynx population booms, but then that causes the hare population to crash, which eventually leads to a decline in the lynx population, and the cycle starts again.
Exactly.
Yeah.
That's a classic example of density-dependent regulation.
Okay, so competition and predation are two big ones. Are there any other density-dependent factors we should know about?
Yes, disease transmission is another important one. In crowded populations where individuals are in close contact, diseases can spread much more easily.
Makes sense. It's like if one person gets the flu in a crowded room, it's more likely to spread to others.
Exactly. Our source mentions an example with bee eaters, those colorful birds. When they nest in dense colonies, diseases can spread rapidly among the chicks.
Okay, so crowding increases the risk of disease outbreaks.
Right. And then there's parasitism, which is similar to disease in that it often spreads more easily in dense populations.
Parasitism, meaning one organism benefits at the expense of another.
Yep, like ticks or fleas on animals or intestinal worms. If there are a lot of hosts packed together, it's easier for those parasites to find new hosts and spread.
Okay, so crowding makes it easier for parasites to thrive. Anything else?
Yes, territorial behavior can also be influenced by population density.
Territorial behavior, meaning animals defending their space, right?
Exactly. And in crowded conditions, the competition for territory can get really intense, which can lead to more aggression and stress.
Well, like more fights breaking out between animals defending their turf.
Right. And that stress can actually have negative effects on their survival and reproduction.
Ah, so it's like a built-in mechanism to prevent overcrowding.
Precisely. All these density-dependent factors work together to keep population size in check.
Got it. Now, let's shift gears a bit and talk about population growth curves. What are those all about?
Population growth curves are basically visual representations of how a population size changes over time.
Okay, so it's like a graph showing population size on one axis and time on the other.
Exactly. And there are two main types of growth curves that we often see: exponential growth and sigmoid growth.
Let's start with exponential growth. That sounds pretty dramatic.
It is. Exponential growth basically means that the population is growing at an accelerating rate.
So like it starts off slow, but then it gets faster and faster.
Yep. And when you plot it on a graph, it creates a J-shaped curve.
Okay, J-shaped, I can visualize that. And why does this happen?
Exponential growth usually happens when a population has access to unlimited resources and ideal conditions, so there are no limiting factors holding it back.
So like if they have all the food, water, and space they need, they can just keep reproducing and growing.
Exactly. It's like, imagine a bacteria population in a petri dish with plenty of nutrients.
They'll just multiply like crazy, right?
Yep. But this kind of growth can't go on forever. Eventually, resources will start to become limited.
Right, the petri dish will run out of space and nutrients.
Exactly. And that's where sigmoid growth comes in.
Sigmoid growth, that sounds less dramatic.
It is. Sigmoid growth is more realistic because it takes into account the fact that resources are limited.
Okay, so how does it work?
Sigmoid growth starts off like exponential growth with a rapid increase in population size.
But then something changes.
Right. As the population grows and approaches the carrying capacity of the environment, the growth rate starts to slow down.
Ah, because those limiting factors start to kick in.
Exactly. There's not enough food or space for everyone, so the growth slows down, and eventually the population levels off around the carrying capacity.
Okay, so instead of a J shape, we get a different shape.
Right. Sigmoid growth curves have a characteristic S shape. They start off steep, then flatten out as the population reaches carrying capacity.
S shape for sigmoid, I can remember that.
Our sources give the example of the Antarctic fur seal population. They were hunted almost to extinction, but then they were protected and their population rebounded rapidly.
So they went through a period of exponential growth.
Right, but as they approached the carrying capacity of their environment, their growth slowed down and their population stabilized.
Okay, so that's a real-world example of sigmoid growth.
Exactly. And scientists can actually study these growth patterns in the lab using model organisms.
Model organisms meaning?
Organisms that are easy to work with in a laboratory setting, like yeast, fruit flies, or certain types of plants.
Okay, so they use these sampling organisms to study basic principles of population growth.
Right. They can manipulate the conditions in the lab, control the resources available, and see how that affects population growth.
Makes sense. It's like a simplified way to understand the complexities of real-world populations.
Precisely. Now, so far, we've focused on how populations interact with their environment, but let's not forget about the interactions that happen between individuals within a population.
Oh, yeah, right. Like competition for mates or cooperation in raising offspring.
Exactly. Those are examples of intraspecific relationships, interactions within the same species.
Intraspecific, meaning within the species. Okay, I'm starting to get the lingo.
And these interactions can be just as important as the interactions between different species.
So what are the main types of intraspecific relationships?
The two big ones are competition and cooperation.
Okay, we've talked about competition before in the context of density-dependent factors.
Right. Intraspecific competition happens when individuals of the same species are vying for the same limited resources.
So they're basically competing with their own kind for food, water, space, mates, all that good stuff.
Exactly. Our source mentions the example of male red deer fighting for access to females during mating season.
Oh yeah, I've seen those nature documentaries where the stags lock antlers and battle it out.
That's classic interspecific competition. And then there's the example of robins defending their territories from other robins.
So they're competing for space and the resources within that space.
Exactly. They need to secure enough food and nesting sites for themselves and their offspring.
Okay, so competition is all about, like, fighting for your share of the pie.
Right. But on the flip side, we also see a lot of cooperation within species.
Cooperation, meaning individuals working together.
Exactly. And this can take many forms, from group hunting to collective defense to resource sharing.
Okay, give me some examples.
Sure. Think about orcas, those killer whales. They often hunt in pods, working together to take down large prey like seals or even whales.
Oh, wow. So they coordinate their attacks.
Yep. By working together, they can take down prey that would be too big or too difficult for a single orca to handle.
That's pretty amazing. What other examples of cooperation are there?
Well, many animals engage in collective defense to protect themselves from predators.
So like a herd of zebras forming a circle to protect their young from lions.
Exactly. By sticking together, they make it more difficult for predators to isolate and attack individuals.
Safety in numbers.
Right. And then there's resource sharing, which can be as simple as two individuals sharing a food source or helping each other to find food.
Okay, so cooperation can be about sharing the wealth.
Yep. And it can also involve things like mutual grooming, where animals help each other to stay clean and healthy.
Like monkeys picking bugs out of each other's fur.
Exactly. And that grooming also strengthens social bonds within the group.
Okay, so cooperation is all about working together for mutual benefit.
Exactly. And it can be just as important as competition in shaping the dynamics of a population.
Got it. Now, let's zoom out a bit and talk about communities. What exactly is an ecological community?
An ecological community is basically all the interacting populations of different species that live together in a particular ecosystem.
So it's like all the living things in a certain area, the plants, the animals, the fungi, the bacteria, everything.
Exactly. And the key here is that they're interacting.
Meaning they're affecting each other in some way?
Right. They might be competing for resources, preying on each other, or even cooperating in certain ways.
Okay, so it's like a complex web of relationships.
Exactly.
Hmm.
And we call those relationships interspecific interactions between different species.
Interspecific, meaning between species.
Yeah.
Got it. So what are some of the main types of interspecific interactions?
Well, one that we've already talked about is predation.
Right, one species killing and eating another, like the lynx and the hare.
Exactly. And predation plays a huge role in shaping communities because it can control the size of prey populations and influence which species are present.
Okay, so it's a powerful force. What else?
Another important interaction is competition, but this time it's interspecific competition between different species.
So it's like different teams competing for the same resources.
Exactly. And this can happen for all sorts of things: food, water, space, nesting sites, sunlight, you name it.
Okay, so competition can be pretty fierce between different species.
It can. Our source gives the example of native bee-eaters and introduced starlings competing for nesting cavities in trees.
Ah, so they both need the same type of nesting site.
Right. And unfortunately, the starlings are often more aggressive and they outcompete the native bee-eaters.
So the starlings are kind of like bullies in this case.
You could say that. And this competition can have negative impacts on the native bird populations.
Okay, so competition can be a problem when one species is more dominant.
Right. But not all interspecific interactions are negative.
Oh, right, like mutualism.
Exactly. Mutualism is a win-win situation where both species benefit from the interaction.
Okay, give me some examples of mutualism.
Sure. One classic example is the relationship between flowering plants and their pollinators.
Right, bees and butterflies visiting flowers to get nectar.
Exactly. The plants benefit because the pollinators help them to reproduce by spreading their pollen, and the pollinators benefit because they get food from the nectar and pollen.
So it's a perfect partnership.
It is, and there are tons of other examples of mutualism in nature.
Like what?
Well, our source mentions the relationship between legumes and nitrogen-fixing bacteria.
Legumes, those are plants like beans and peas, right?
Yep. And they have these special nodules on their roots that house nitrogen-fixing bacteria.
Nitrogen fixing, what does that mean?
It means that the bacteria can convert atmospheric nitrogen into a form that plants can use.
Ah, so the bacteria are providing a valuable service to the plants.
Exactly. Nitrogen is an essential nutrient for plant growth. And in return, the plants provide the bacteria with sugars and a safe place to live.
Okay, so they're both getting something out of the deal.
Yep. Another cool example is the relationship between mycorrhizae and plant roots.
Mycorrhizae, what are those?
Mycorrhizae are basically fungi that form a symbiotic relationship with plant roots.
Symbiotic, meaning they live together in close association.
Right, and in this case, the fungi help the plants to absorb water and nutrients from the soil more efficiently.
So the fungi are like little helpers for the plant roots.
Exactly. And in return, the plants provide the fungi with sugars from photosynthesis.
Okay, so another win-win situation.
Yep. Mutualism is all about cooperation and mutual benefit.
Now, on the flip side, we have parasitism.
Right. Parasitism is where one species benefits and the other is harmed.
So it's a one-sided relationship.
Yep. The parasite benefits by getting food or shelter from the host, but the host is usually weakened or harmed in the process.
Okay, so it's like a freeloader situation.
Haha, kind of. Our source mentions the example of the avian vampire fly, which feeds on the blood of nestling birds in the Galapagos.
Vampire fly, that sounds pretty creepy.
It is. And there are tons of other examples of parasites, like tapeworms in the intestines of animals, or mistletoe plants that steal nutrients from trees.
Okay, so parasites can be pretty sneaky.
They can. And then there's another term, pathogenicity, which is closely related to parasitism.
Pathogenicity.
It basically means the ability of an organism to cause disease.
Ah, so pathogens are like disease-causing agents.
Exactly. And many parasites are also pathogens.
So like a virus that makes you sick is a pathogen.
Right. And pathogens can have a huge impact on populations, especially if they cause widespread outbreaks.
Okay, so we've covered predation, competition, mutualism, and parasitism. Are there any other types of interspecific interactions?
Yeah, our source mentions a few others, but they're not as common.
Like what?
Well, there's commensalism, where one species benefits and the other is neither helped nor harmed.
So it's like a neutral relationship.
Exactly. Our source gives the example of hermit crabs using empty shells left behind by other animals.
Ah, so the crab gets a home and the original owner of the shell doesn't care because they're already gone.
Right. And then there's amensalism, which is kind of the opposite.
Opposite in what way?
In amensalism, one species is harmed and the other is unaffected.
So it's like a negative impact, but the other species doesn't even notice.
Exactly. The source gives the example of the penicillium mold, which produces penicillin.
Penicillin, the antibiotic.
Yep. Penicillin kills bacteria,
Yeah.
but the mold itself isn't affected.
Okay, so the bacteria are harmed, but the mole is just doing its thing.
Right. And finally, there's neutralism, which is basically when two species have no direct interaction or effect on each other.
So they're just kind of coexisting?
Exactly. It's like two ships passing in the night.
Yeah.
Got it. Now, our notes bring up the issue of invasive species. What are those all about?
Invasive species are species that are introduced to a new environment where they don't naturally occur.
So they're kind of like outsiders.
Right. And the problem is that they can often outcompete the native species, causing all sorts of ecological problems.
Why are they so successful?
Well, for one thing, they often don't have any natural predators or diseases in their new environment.
So there's nothing to keep their population in check.
Exactly. And they might also be very adaptable.
Meaning that they would be able to cope with a wide range of different environmental effects. Okay, guys, I hope this has helped. Remember to keep on studying hard and look at the environment around you. Remember to like and subscribe. Take care. Bye-bye.